Okay, this video is going to talk about processing steps moving EEG data into event-related potentials, or ERPs. This presentation was made possible by materials from Pursue. You should check them out, pursueerp.com. So the learning goals for this particular video, you should be able to describe the general pre-processing steps and explain the purpose of each processing step. It's pretty simple, a lot of steps though, so long video. So overall, the goals of pre-processing our data are to reduce the noise, and in particular, we're going to do some filtering and artifact rejection, which will accomplish that goal. We're going to transform the data so it can be compared across trials and conditions, and we'll be doing baseline correction, some re-referencing to do that. We're going to extract segments of the EEG data so that we can average them together. These are called epochs. And actually when we create the average, we're going to do the averaging process to create the ERP. So a couple things, background things to know about pre-processing EEG data. There is something that people refer to as the pre-processing pipeline. This just refers to the sequence of steps that get to the data that are actually analyzed. These steps are going to differ across labs and across areas. It depends on equipment, ERP components that are targeted. There's some personal preferences that get in there for sure. The field is actually getting better at sharing these steps, talking about them, fully disclosing the pipeline. You should hopefully start to see this in recent articles where people are fully disclosing each of the steps that they used for their data treatment. Each step has some pros and cons, and some of them have a lot of technical information. This is just going to provide you with a little bit of background. Obviously, there's some other sources that you could dive deeper into any one of these areas should you want to explore a little bit further. Now, I just want to talk a little bit about pre-processing versus processing, because I always refer to this as processing. So a couple definitions for you. The pre-processing, we don't do a lot of data dropping. I'm pretty conservative in that regard, and I don't like to remove data. So I try to keep as much of it as possible by correcting it. So I typically refer to this as data processing rather than pre-processing. It's a small semantic difference, but I just wanted you to know that as we're going through this video. When I talk about processing, some other people are going to call it pre-processing. Once again, this is just a conceptual overview. The protocol that we have in our lab has step-by-step instructions for how to do all of these steps in MATLAB, EEG Lab, ERP Lab. And also there's companion videos that sort of show you these steps. But here we're just going to talk about the conceptual basis of this. So our steps, we have 7 different steps that we're going to go through, and we'll revisit each one of these steps at each phase to explain what they are and what we're doing with each one of these. So the first step, and maybe I could even put in, the first one would be to import the data file, but we'll just sort of assume that that's already going to take place. You have it in MATLAB. First thing we're going to do is downsample. So we sample at a really high rate, 2,048 samples per second. And we do this on our amplifier so that we capture all of the event triggers exactly as they're happening. But that's a really high sampling rate. We don't need that much data. We quickly downsample to 256 hertz to create smaller data files. This also speeds up our processing stages and such with less data. Now remember, anytime you're downsampling, you do want to be concerned about aliasing, but remember, bandwidth we're kind of interested in, and up to about 30 hertz or so. So even if we multiplied 30 hertz by 3, right, to avoid aliasing, we're still at 90 hertz, and you can see that we're well above that, so we're not going to have that aliasing issue that we talked about in a previous video. So the second step is to filter the data. And technically you can filter based on frequency, or you can filter based on time. Now, in my lab, we just do frequency filtering. We don't really do time filters. I will briefly talk about one just as an example so that you sort of know what it is. EEG can be measured in frequencies, which are cycles per second. So sine wave cycles per second. The measure of that is hertz. So 1 hertz is 1 cycle per second, for example. So when we do a step of artifact detection, we're going to get a frequency spectrum like what you can see here in this bottom graph here with this red line. And on the x-axis you have frequency, and then you have power over here on the y-axis. And so in this graph you can see there's a little bit of a bump around, say, 12 hertz or so. And the point here is that this doesn't mean that you're going to have a 12 hertz sinusoidal wave in your EEG data, because your EEG data is actually a mixture of many signals. And in fact, you can see there's some low frequency activity that would also be mixed in as well. But it does point out that we have different frequencies of activity sort of smashed together in our waveforms. So now, why would we filter? Because some of these higher frequencies or lower frequencies are actually artifacts. So filtering can actually help remove some artifacts. So what we're trying to do is isolate our brain-only activity, right? So roughly 0.1 hertz to 30 hertz, right? And when we do this, when we do some filtering, not only will it isolate that brain activity, but it'll make the data a little bit smoother, a little bit easier to sort of see. And at the same time, what we're doing is we're reducing 60 cycle or 60 hertz activity. This is the frequencies of the electrical activity in electrical wires. So we want to reduce that. That could be a contamination. We're going to reduce movement or EMG. So these would be higher frequency waveforms that we can record, but movements are a big artifact. And we also want to remove those low frequency artifacts like skin potentials and electrode drift, etc. So filtering helps us get cleaner data in general. What we mean by cleaner data is isolating brain data. So frequency filters are kind of like coffee filters, right? They're going to allow certain things to pass through and keep out other things, right? So with these filters, and we'll see some examples in a minute, you're going to set a threshold that will allow some frequencies to pass and block other frequencies. So there's a couple different kinds of filters, and we're going to talk about examples of each one and what they do. You can see some filtered versus unfiltered data, and the filtered data is a little bit smoother, and that makes data analysis a little bit better. So types of filters, we have a low pass filter. This is when you set a threshold. These terms are kind of counterintuitive, but you're setting a threshold and you're allowing frequencies to pass below whatever your threshold is, right? So everything below that threshold is going to pass through. So what it does is it removes higher frequency noise. So let's say, for example, we're going to use 30 hertz as our low pass filter. And we do this. We actually do a band pass filter, but 30 hertz is what we use for the low pass part of that. And what that means, as you can see in the slide, is that they're defined by their half amplitude cutoff. So 30 hertz means that half, this is the gain or the magnification of that particular frequency, would be at half or half of the amplification. And so that doesn't mean that everything right above 30 hertz is cut off. It just means that it's getting less amplified, and the further out you go, there's less and less signal until eventually it's zero, where there is no passing of those higher frequency amplitudes. And so remember, this is going to reduce higher frequency things, pretty much like movements. It's going to get rid of a lot of the movements by filtering. So again, this is an example of a low pass filter. So you can sort of see in that example that we saw a moment ago how it's a little bit smoother with this particular low frequency, or with how it's a little bit smoother with this low pass filter applied. Now, high pass filters, on the other hand, are only going to pass frequencies that are higher than whatever threshold you set, which is why I said this is counterintuitive, right? So whatever value you set, frequencies higher than that value are going to pass through. And so they are going to remove slow changes or low frequency kinds of contamination like skin potentials or drift. Now, one thing to keep in mind is if you, and we'll see an example of this in just a second, if you use aggressive high pass filters, it can distort your ERP in particular. So for example, here is a high pass filter. So remember, it's sort of defined the same way. When your gain or your amplification is at 0.5, that's the value. So for example, if you used a high pass filter with 2.5 hertz, what that means is at 2.5 hertz, the gain is 0.5, right? It doesn't mean that nothing below 0.25 hertz is going to be passed through. It will, but just at a lower gain. Now, over here on the right, you're going to see what happens when you increase or use higher and higher, what I would say more aggressive high pass filters would be. So you see the black line is the unfiltered data, and then red is 0.01, blue is 0.1. Now, just for reference, we use 0.1 for our band pass filter and so on. But if you used even just 1 hertz versus 0.1, you can see a dramatic difference in the ERP amplitude there. And potential distortion there. So you want to be wary of that and pay attention to that when you're reading papers. What are their high-pass filters if they're using them, or bandpass filter settings? Now, a bandpass filter is really just smashing a low and high-pass filter together, right? So you're just specifying a range. So you're doing it in one pass, but you're filtering everything, above a lower edge frequency or below an upper edge frequency. And that's what we call a bandpass filter. Now, other filters that are sometimes used, we don't really use them very much. But notch filters, so if you had a particular set of frequencies, so for example, if you're recording in an environment that has a lot of electrical noise, like lights or from electricity in the walls and such, you might get a lot of 60 cycle contamination, in which case you could use a notch filter for 60 cycle, and really just is around that particular frequency. It tries to target a certain frequency for removal, and that's called a notch filter. Now, we've been talking about frequency filters. You can filter over the time domain. So just as a reminder, time here, we're talking about, x-axis would be time and y-axis would be the amplitude. We don't use these, but you might come across these. And so an example of a time domain filter is a moving average filter. And essentially what they do is they take the average of certain points over time and then replot the ERP with the average over those points in time, so that it kind of the window keeps moving, hence the name moving average or moving window average, and you get a smoother waveform, right? So it's a way to sort of smooth out, get rid of those jagged peaks and valleys, and some people use them. So some filtering caveats, in particular, pay attention to really steep roll-offs or narrow bands, because that's going to flatten or potentially distort your data. And so what we mean by a roll-off is this slope of this line here, how quickly it's diminishing the gain of any frequencies that sort of passed by. And remember, gain is the amplification factor, right? So really steep or narrow bands are going to potentially distort the data. So you want to pay attention to that if you're reading papers, if you sort of see that. Step 3, after we filter, we're going to epoch and baseline correct. So remember, our EEG is recorded in continuous signal, and you'll see that in our data files. And then we have various event codes, markers that indicate both the appearance of stimuli and responses, etc. And here you see an example there. It can have a number, and then we change it to actually have a code to identify the trial type. And so an epoch is a time window around some sort of event marker. So it's a period of time that is predetermined or should be predetermined. And epoching means that what you're doing is extracting time around this event from your continuous EEG segments, and you basically just line up these epochs next to one another in a file. But you're pulling out targeted data and leaving behind the other data that you're less interested in, like stuff that's happening in between trial. So the epoch starts at time 0 is when, say, like for example, a stimulus appears on the screen. That's most prototypical, but you could have response locked ERPs, in which case time 0 would be when the response happens. The window starts time 0 when the stimulus is on the screen and extends for some period of time. And this I always determine in advance. And so if I'm doing recognition memory studies, I will use about 1500 milliseconds, because that's plenty of time. Pretty much everything that we're interested in happens up to, say, about maybe a second or so. And so the extra 500 milliseconds is just make sure that you're capturing everything you want to capture. If we're doing source memory, those decisions take a little bit longer, so we would push it out to maybe like 2.5 seconds or 2500 milliseconds, use a bigger window. It also includes, the epoch also includes some sort of baseline period. And again, we typically use 200 milliseconds before the event code, and what we're going to use that period for is to baseline correct our ERPs, and we'll get into that in just a second. So you want to design your experiment with the epoch length in mind to prevent overlapping epochs. So let's say, for example, that we use a little bit of a 200 millisecond baseline, 1500 millisecond time window. And you can see if we design our experiment to have a long enough intertrial interval, we wouldn't have overlapping epochs at all. However, if we used a much larger window, say we used like a 3 or 4 second window, you could see that your epochs here would overlap. And that's not very good, right? Because then they're going to start overlapping and you're going to be piecing bits and stuff together. And you don't really want that. You want to avoid that if you can. So the key here is to have a large enough intertrial interval, that's called an ITI, to make sure that you're accounting for not only your epoch length, but a little extra time before the next trial begins to prevent those overlapping epochs later on when you do data analysis. So baseline correction. So what we're doing with baseline correction is we're subtracting the time right before the stimulus to sort of zero out or recenter our waveforms. So as you can see here, the red line would be what would be recorded, and then the green line is the baseline corrected version of that form. And the reason we're doing this, adjust for any kind of voltage offset or drift. Even between electrodes, the EEG will have different values, and you want to re-zero everything right at time zero when your event is happening to see. We're interested in the changes from time zero, essentially, or slightly before time zero. That's what's interesting, is what are the changes in response to that stimulus, not the overall value when the stimulus actually happens. So this slide shows you how one electrode could drift over time, over many different trials, and the baseline correction would sort of adjust for that. It would put them all sort of on the even scale there. In other words, the change over time is not what we're interested in. We're interested in the change relative to the presentation of the event. One thing to keep in mind, particularly when you're evaluating your own data or reading articles, is look at the baseline. And if the baseline itself is pretty flat, then that's pretty good. If there's differences in the baseline across conditions, that would be a particular concern. Now, it may go up or down depending on what's happening right before that stimulus, so that could differ, and it could be okay depending on the situation, but it's something that you want to look at and pay attention to sort of see, is there a baseline issue that is going on with that particular paper. Again, some considerations. Use a adequate intertrial interval so that you can basically meet the assumption that the activity is just offset voltage. It's not overlap from a previous trial that could impact things. The other thing is you don't want to use too long a baseline period because you're going to have more artifact rejection, and again, you could get some contamination from events that are happening, right? Mental events that are happening before the stimulus even appears. Okay, in our fourth step, we're going to do some artifact rejection, and this means that we're going to do something called independent components analysis, or ICA. And so the idea here is that artifacts are separated by the ICA. We're going to evaluate the individual components, and then we keep those that are produced by the brain and remove those that are artifact, not brain. So I just want to talk about different types of artifacts. So one big source of artifacts are ocular artifacts, or blinks. And so if you remember from our previous videos, the cornea is positively charged with respect to the back of the eye, the retina, and your eye movements or blinks can create lots of activity that spread across the scalp. And so blinks are a source of this. And here you can see a VEOG signal indicating that the person closed their eyelid and created a blink, and then that spreads across the electrodes. It's much larger at the frontal electrodes, and it diminishes in amplitude as it spreads across the scalp. Now, one of the things that we can do is we can reduce blinking. Some people ask their participants not to blink. We don't really do that, but we do encourage them to blink in between trials. I don't want them necessarily thinking a lot about blinking because I think that can introduce other problems in the experimental design. But we definitely encourage them to blink right after they respond. And we also want to make sure that our ocular electrodes are not too close to their eye, because if you get it too close to the eye, it's going to cause it to irritate the eye and blink a lot more. So you want to make sure that there's plenty of space and that it's not actually causing them to blink a little bit more. Now, the other source of ocular contamination is movements. They could be vertical eye blinks. Here you see horizontal eye blinks, and so shifting of your eyes left to right. So any eye movements are called saccades, of course, and we could reduce eye movements, both vertical and horizontal. We do this in our lab by sort of encouraging to memorize their response pad and also having it placed so that they're not moving their eyes too much to see which keys to press. So when you give instructions, that's part of our instructions, is to encourage them to sort of memorize which key they're supposed to be pressing to decrease the amount of ocular artifacts. We can also get some drift. These are slow voltage shifts that can result from skin potentials, sweating. You could have the participant disturb the electrodes by moving or touching the cap, or changes in the impedance. So the electrolyte could dry out or it could settle. So there was a period of time in my lab where students weren't filling the electrodes after they got the impedance low, and we had all kinds of electrode drift problems. I then discovered this and encouraged them to fill up the electrodes after the impedance is below 5 kilo ohms, which is our threshold, and then all of a sudden our electrode drift problems disappeared. So what I think was happening is that the electrolyte was sort of settling, and as it settled, that caused greater impedance and more electrode drift. Filling those electrodes before you're done prevents that from happening. You can also have what's called blocking. This is when the signal gets out of range and clips off the signal, so it becomes just sort of this flat line. It doesn't read any higher values. We don't really have that problem with our current amplifier. It's really good at still recording signal no matter what the situation is, but we still sort of look for it. And there are circumstances where we want to detect sort of no signal or flatline. And we could have no signal if an electrode became unplugged, for example, or something went wrong with the amplifier and that kind of thing. So we do want to look for it, but it's pretty rare for us. EMG is movements, a lot of times from jaw clenching, but also physical movements of the legs, feet, whatever, hands. You'll see that as a higher frequency activity like you see over there. And we could also have heart rate activity. This is also pretty rare to see, but it does happen, where you can sort of see some bleed over EKG. A heart generates a lot of electrical activity, so we can actually pick that up. In addition to what Steve Luck calls commonly recorded artifactual components, or CRAP. So examples: tapping your foot, bouncing the leg, chewing gum. We want to make sure that they don't have any gum. Talking to oneself, and those kinds of sorts of things. So you want to make care in describing the instructions to try to eliminate those sorts of things. You also want to monitor the person while they're doing the experiment to sort of see if they're doing some of these things. And sometimes you can go in and remind them or let them know if we do have a break in between, and maybe that can decrease some of these other artifacts, but we want to be mindful of that. We know that we have these artifacts. What are our options for dealing with them? Well, one of the things we can do is reduce them, and I just talked about some ways in which we can reduce them. We can encourage them to blink after they respond, memorize the keypad, maybe not move around so much, that kind of thing. So a lot of our instructions reduce the occurrence of the artifacts. Then we can correct. That's where we are right now. In this step, we can estimate the influence of these artifacts and then sort of take them out of our data by still retaining the data. And we're going to use independent components analysis to do this. And then our last option is to actually reject. So if you go through all that process and you still have artifact in it, well, we could just drop that individual trial and eliminate it from our data. But we want to do this very prudently, right? We don't want to just eliminate a ton of our data, because that's not good either. So the artifact correction that we're going to use is independent components analysis. And so this is a really good tool that a lot of people are now using. It's very common to use. And again, so our common artifacts: blinks, eye movements, muscle activity, etc. Just one caveat here: although it's going to detect that, this isn't a perfect, but it works really well, and a lot of people are now using it. I'd say it's the most common thing used these days to eliminate artifact. So just as a background, our EEG is a mix of signals from both brain and from these other sources of noise that we just talked about. We want to remove the non-neural sources of noise and keep the brain signals. That's the basic idea here. So ICA is an algorithm, essentially calculates different sources of signal and separates them. And the good news is that the artifacts are really big relative to the brain, but essentially it'll separate into independent components. So here over on the right, you see IC1, IC2. That's independent component 1, independent component 2, and so on. And we can inspect each one, select which ones are due to artifact, and remove them and have our data corrected, right? So that's what we're going to do. So the idea is you might identify, independent component 1 and 4 are artifact, and so we're going to take them out of the data. So what you would get, your corrected EEG, would just be the mix of independent component 2 and 3. in this simple example. It's more complicated than that in practice, but that's the idea. Now, the other thing that we use is we use this thing called IC label, and IC label is a artificial intelligence that will look at each one of the independent components that get separated in our data, and it'll label it with what it thinks it is. And so our job is to sort of look at them, make sure we agree, and essentially keep them. So it'll label it like you see I here, but it'll also label it other, and we want to inspect for sure ones that are other. There's other training videos, and you'll practice this in the lab to learn how to do this, to how to sort of evaluate this. But we use the artificial intelligence IC label in our decision-making process to decide whether we're going to keep or remove individual signals. After we do artifact rejection with ICA, we run some ERP lab routines to capture additional artifact, anything that survived ICA and artifact correction. Now, the way this works is it's a routine. Each one of these is a routine that will run through, and if it detects the artifact, it's going to reject that trial. So we would actually drop or lose the trials. So this is not the preferred method. This is sort of the last resort kind of method. Now we're going to do three routines in ERP lab. We use the moving window peak to peak, the step function, and the blocking and flat line. And I'll explain what each one of those are. So the moving window detects rapid spikes in your data like those that appear in blanks. Now the specific parameters for the moving window are in the protocol, so there's some numbers that you'll make sure that get put in, but essentially it measures the difference between the highest peak and lowest peak within the window, and the window and the threshold are set by our parameters that we put in. And it's going to mark the trial for rejection if the peak-to-peak amplitude exceeds that threshold, right? That's the idea here. So it just goes through and detects those. The next one that we use is step function, and the step function is really good, as you can sort of see here. Again, you have parameters that you put in that come from the protocol, but it looks for different windows and looks for those different steps, those little, like you can see here, the brown sort of figures that appear in here, these different steps. And so what it would do is it would detect changes that sort of meet that or exceed the values that we set for the step function and identify, in this case, an eye movement. And it's really good for detecting particularly horizontal eye movements. I find that it's really helpful. If you have a lot of that, you'll see a lot of the trials get flagged if they can't be corrected with ICA. So blocking and flatline. So again, I said that we don't have a whole lot of clipped off signals, but if you did, then this algorithm would detect that. Most of the time, we use this to sort of find out if you overcorrect, like if you remove too many components, some of your channels might go to 0 and you might have no signal, in which case you might get a lot of trials that have flatlining in it. So that's where I see it most helpful, is identifying if it's been overcorrected. Prototypically, you won't have any trials that would be flagged because of blocking and flatline. So evaluating your data. So in one of our processing steps, you're going to get this table, a table like the one that you see over here. This is just sort of a prototype. It differs. So the bin number over here, you see 1 through 12, that's going to differ depending on the experiment. And what each bin corresponds with is an ERP of a different type. And so that's going to change from experiment to experiment, but there's some bins that we're going to pay attention to, and maybe some bins that are less important for our research purposes. But the first column you see is the number of trials. So in this case, 42 trials were in that first bin. That's a healthy number, so that's good. And it also, this is the number that was accepted. And you see in the second column the number that were rejected. And in this case, all 42 were accepted, 0 were rejected. Now the number of trials is going to depend on what that bin reflects, the person's response, and whether there were any trials that were flagged by the algorithms, right? So we want numbers that are greater than 14. 14 is our lower threshold. So you're going to check all of the bins, and the relevant bins will be in the protocol. It's not all of them usually. It's usually like 1. It usually alternates. So like 1, 3, 5, 7, etc. And you would look at them and you would see, is the number greater than 14? If the number is greater than 14, then that's good. Bottom line, that's good. The second column, the F2 column where the red arrow is, this shows you the number of trials, and you can see we have a couple of trials here that were flagged by the moving window. So that only 2, That's not a big deal. You can see that 2 trials out of 358 trials were removed. So that's only 0.6% of the data. That's great. That's perfectly fine. Caught a couple trials and removed them based on that moving window. F3 shows for the step function, and this is pretty prototypical. You rarely have one, but sometimes you do. If you do, maybe that suggests that you have some eye movements that you missed during ICA. And as I have here for, if you have a lot of trials in any one of these columns, it suggests maybe you missed something during ICA, and you could always go back and maybe remove a component that you weren't too sure about and try it again and see if that fixes it. Now this last column, blocking and flatline, as I mentioned, if you have a lot of numbers here, it suggests that maybe you overcorrected. Maybe you used too many removed too many components from the ICA and overcorrected. Typically, it would be 0, maybe 1 or 2. That would be perfectly fine. And bottom line is, down here at the bottom, you want to sort of see that, like, are you retaining, are you only losing like maybe 10% of the trials? 10% would actually be a little bit high. Are you somewhere between 0 and 10%? And if you are, and you still have numbers of each of our critical bins that are above 14, then that's probably okay. If not, if the numbers are too low, then we're probably going to replace that participant. That just means that that's not usable data, and we're going to move on. Often what will happen is, if you're actually processing, and this describes your situation, is you would let me know, and then I would go take a look at the data and sort of report back whatever I found, so that you could learn from that particular experience. In step 6, we're going to re-reference. So remember, there's multiple ways that we can reference, and when we're recording online, everything is referenced to the left mastoid as the online reference. We're going to re-reference. We're going to change the reference to the average of the left and right mastoid, and that's because we run the right mastoid as an active channel. It's M2. It has impedance, and you're going to prepare it, etc., and you're going to see it in the EEG. And so we run all of our data through all of these routines, and we end up with an active channel at M2. And at this stage, we'll then re-reference to the average of the left and the right mastoids. And remember, just a refresher, the reference matters a little bit. It's prototypical in memory research to use the average of the left and the right mastoids. References can change things. They can reverse the polarity of the signal. So it's important to sort of use a standardized one, and that's what we do. So the data here it's just a simple, the average mastoid equation. You have your new channel, you take the old channel, and you're going to subtract off the average of the 2 mastoids, and then you have your new reference. Some people would do a common average reference, and in which case what that would mean is you're going to take the old channel minus the average of all of your channels. If you had a 32 channel system like we do, then it would be the average of the activity channels 1 through 32 to give your common average reference. Now, not all channels are re-referenced like our bipolar channels. So our bipolar channels make, in particular, eye artifacts easier to detect. So we record VEOG and HEOG as a bipolar channel. What that means is that each channel is referenced to the other electrode. So like VEOG, so they're not using the left mastoid as a reference. Instead, the upper VEOG is referenced to the lower VEOG, right? And the left HEOG is referenced to the right EOG. Right? So that's what that means, that bipolar reference. And this just enables us to detect better artifacts. Now, the important practical bit for this is the impedance appears as a pair on our screen from our amplifier. And you're going to lower these. It gives an individual reading for the upper and the lower and the left and the right. But really, they're lowered as a pair. So if you're having trouble lowering the impedance on one of them, work the impedance on the other one, because I find that sometimes helps quite a bit, and think of them as a pair, not necessarily as an individual, or use that individual value as sort of take it with a grain of salt. Let's put it that way. Okay, our last step is to average the ERPs. Now remember, individual ERP trials are fairly noisy, and so what we're going to do is we're going to average across like signals to reduce the noise and extract the common signal, or the event-related potential, right? The activity that's commonly elicited by that particular event. Remember, the pre-processing removes a lot of these large artifact noise, but we're trying to record these really tiny signal changes in response to our stimuli, so averaging is going to be our last line of defense to try to extract those tiny signals. So what we're going to do is handle a simple example of an N400-like experiment. So just to illustrate this purpose, let's suppose that you hear different kinds of sentences. So like you hear a sentence, the pizza was too hot to eat. In that sentence, the word eat is congruent. It finishes the sentence is congruent with the meaning of the sentence, right? But you contrast that with the pizza was too hot to cry. Cry is incongruent with the meaning of that sentence. And we just keep repeating these kinds of sentences, either congruent or incongruent over time. And we're going to record the EEG, of course, right? And we have different trials which correspond to each of these two different trial types. Now, what we're going to do is we're going to look at the ERP and we're going to average. So we're going to take our too hot to eat, and so we get an ERP for congruent endings, and then we're going to average those that are incongruent, and we end up with a different ERP, right? And if we've done a good job with all of the signal extraction, they're going to be nice and smooth like this graph, and you're going to see a nice N400. Remember, negative is up in this particular graph. You're going to see a nice N400 for the incongruent sentences, right? So that's the idea behind averaging. So how many trials do we include in average? You saw on that earlier slide, our lower number is 14. That's a lower number. The greater, the better, right? That's our minimum number, let's say. So the ERP noise varies with the number of trials that you use. More is better, better signal to noise ratio. Now, we try to design our studies so that we have a fairly high number of trials available for averaging, but that depends on how many conditions we have, and that can also affect how long the study is. There's only so much time we can expect someone to be in the study and complete it. So sometimes we have to make some decisions about that. And so again, if you think about this in terms of noise, our lower end is somewhere between 9 and 16, right? 14 is our lower end. We really like to be around 25 to 36 in this neighborhood consistently. And if we can get higher, depending on the study, then we like that, but for memory studies, that can be pretty difficult, particularly if you're interested in more than 2 conditions. The last part of data processing is to graph your data. So there's instructions for how to do this, and we don't reject subjects based on their graphs, but I just like you to get practice plotting and also looking at the data, and also you're kind of looking at it for any anomalies that may remain. So like in this particular example, you're seeing the ERPs at the different electrodes. They all look pretty good. It looks like we have a good signal here. You can look at your occipital electrodes to sort of see, do you have a signal which is related to visually presenting the stimuli, since all of our studies are visual presentations. And, is that clearly marked in the ERPs? That is, are we getting signal? That's sort of the first question I ask. And then the next question I ask when looking at these is there any drift or big ugly signal that looks like some kind of artifact that still remains in the data? If so, we might want to go back and look at it. You could always make a note and I could look at it, and then we could go back and look at the data processing and sort of see if there's something that we missed during the processing. But you want to just sort of eyeball it to sort of see, does it look good? And this takes some practice, but we'll practice that as well, and you'll sort of get a sense of what's okay and acceptable and what's not okay and what may need some more attention. All right, so that takes you through all of the steps for our processing.